All right, so step 2 of protein synthesis, translation. But before we get into that, let's just make sure we're all on the same page and just go over some vocab. So if I ever talk about your genetic code, or you've heard that before, all we're talking about is a code of instructions for how to make proteins. That is what your genetic code is, your DNA. Code of instructions for how to make proteins. So next term, codon. A codon is a set of 3 nucleotides on the mRNA. So when we're talking, when we say code of instructions, this whole section, translation, is about what exactly we mean by that. And so there are 20 amino acids that make up proteins, that are the building blocks of proteins. And so each amino acid will have a specific sequence of mRNA nucleotides that will code for that amino acid. So for example, if we're looking right here at this codon right here, UCA, only one amino acid will be coded for if we encounter the sequence of UCA. And so actually I can tell you which amino acid that would be. We're going to get here. So we said it was ACU. Oh gosh, UCU. Okay, if it was UCU, sorry, I forgot. UCU, it would be the amino acid serine here. Okay, so there we go. Very specific sequences. Oh, UCA, dang it. Okay, so very specific sequences of nucleotides code for specific proteins, and so this is the language that we use to actually build a protein. It's kind of like if you've ever heard of like binary or like hexadecimal systems. Those of you like super nerds out there, it's basically a language that computers speak, like in zeros and ones, and those sequences of zeros and ones correspond to instructions for that computer to follow. And so this is exactly the same thing. So it's just a very specific way of translating information. And it's kind of encrypted in a way. Okay, so the second term I want you to know is anticodon. And so an anticodon is found on a tRNA molecule, so transfer RNA molecule. So remember that little funny looking green thing in the simulation we looked at last week? That is what this little guy here is. And so the anticodon is found on the bottom along here, and it is just a complementary strand sequence of 3 nucleotides found on the transfer RNA molecule. And so if we were to match up this complementary sequence to its complementary strand on the mRNA, or to its complementary codon, then this tRNA will bring its specific amino acid and will add it into the chain of amino acids at the ribosome. Okay, that was a little bit confusing. Don't worry about it too much. The simulation it really helps. It will help a lot, and we'll practice it. But essentially, I just want you to think codon on the mRNA, 3 nucleotides, and there is a very specific anticodon found on tRNA molecules that will match up to the codon on the mRNA, and that is how we read those sequences and bring the correct amino acid that those instructions are calling for. And so then, yes, like I just said, the amino acids are attached here up at the top. And remember, amino acids are the monomers or the building blocks for making proteins. And when we link those amino acids together with something called a peptide bond, which is just a really strong covalent bond, remember, atoms are sharing electrons because they want to fulfill they want to have a complete outer shell of electrons. If you have no idea what I'm talking about right now, you should probably go back and watch Chemistry of Life videos. It was a Unit 1, Concept 2. You should rewatch that because that'll be really helpful. Okay, so translation, the actual steps. Let's get into it. So the purpose of translation, again, is to read and follow the instructions carried on the mRNA to make a protein. And we say that translation occurs in the cytoplasm, specifically at the ribosomes, but ribosomes are they're located on the rough ER, or they're just kind of floating freely. And translation starts with mRNA, so mRNA just left the nucleus and they exited through something called these nuclear pores. And basically, if you just think of like the pores on your face as little tiny holes, that's what it kind of is on that nuclear membrane. So there are these very tiny nuclear pores, holes, that are just big enough for single-stranded mRNA molecules to exit through. But they're a little too small for double-stranded DNA or for chromosomes, definitely not chromosomes. Chromosomes cannot pass through those pores. So they're just big enough for mRNA. And then we say that we end with a protein. Our final product is our protein because that is the whole point of protein synthesis, the whole point of cellular organelles working together. Hashtag proteins matter. So, step one, the mRNA, so it's already left the nucleus and it attaches to a ribosome. Next up, the ribosome reads the mRNA codons. Remember, those are just 3 RNA nucleotides. And then just a little aside. At any given time, tRNA or transfer RNA molecules are just floating around the cytoplasm carrying their specific amino acid that goes along with their anticodon. So the same amino acid will always be bonded to the tRNA that has its matching anticodon. So that it's not just like a free-for-all. But just so that you understand, at any given time, these tRNA molecules are just kind of hanging out. That's kind of like what cells are in general. They're just kind of like this mush pot, this kind of soup. I mean, they're not actually like soup, but that's what I kind of want you to think of it as. It's just like a soup filled with like tRNA, filled with like water, filled with like sodium and like potassium and like all these different random substances can always be found within the cytoplasm. Okay. So, because these molecules are just kind of always hanging out, they act like taxis to drop off the amino acids that match with the current codon. So, the ribosome is reading those mRNA sequences, right? Reading those sequences, and then the tRNA is just kind of floating, and it just happens upon the ribosome, and it goes like, hey, that's my BFF. That's my codon for my anticodon. It has to do with like chemical bonding and just basically there's an attraction there. And so when that tRNA taxi just kind of happens upon the ribosome, it's like, oh hey, I didn't realize you were here. Attaches, and remember only the one that has that correct complementary anticodon for the codon will attach and drop off that amino acid. And so tRNAs will continue to drop off amino acids, and the ribosome will bind those amino acids together with peptide bonds, some just really strong covalent bonds. And then when the stop codon is reached, so there is a specific sequence of nucleotides that codes for a stop, so the ribosome will then stop translating. The ribosome will then release the completed protein, and that cycle is complete for that one ribosome. So, translation is all about interpreting the RNA message into a protein. So here we can kind of see the picture of like what's happening. We have our tRNA molecule right here. We have our mRNA codon. So our mRNA codon here would be AUG. The tRNA anticodon would be UAC, and it drops off this amino acid, whichever one that codes for, and the ribosome will then link it together. If it's the first one, it has something to link to. It just kind of hangs out for a second. But then, if we're on our second one, let's say over here, because we're looking at another tRNA molecule with its anticodon bonding to its complementary codon. Then the ribosome would link these 2 amino acids together in a chain, and then this tRNA molecule would leave, and we'd have our chain right here. And then a third tRNA molecule would come, drop off, connect with its complementary codon, and then the ribosome would link these 3 amino acids together, so we would have 1, 2. We'd have this nice chain of 3 amino acids, and then this one would go bye-bye. And then we'd have another one come in and then drop off its amino acid, and then we would have a sequence of 4 right here. Okay, and so this just happens as the ribosome just moves along and just travels along the strand, attracting tRNA molecules to come in, drop off their packages, and then leave. Okay, so if you were to play the role of a ribosome or of a tRNA molecule, you are going to need this genetic code. So if you wanted to look up which specific codon matches for which amino acid, this is what you would do. So let's say we were looking at there's the start codon. There's a start codon and a stop codon. The universal start codon is AUG. And so let's look at how to use this chart. So we would go over here. So the first nucleotide is A, so we'd start in this box. The second one is U. We would go down here. to this box, and then the third one is G, which is in this box. And so the codon AUG codes for methionine. Remember, there's 20 amino acids. It's just the name of an amino acid. And so that amino acid found on its specific tRNA molecule would be attracted to the ribosome and would drop off its amino acid. Okay, but let's say we have the codon, we encounter the codon CAA. So we would start here in this corner, we go C, A, and we go A, and we would see that this is glutamine, so another amino acid. And if you have not noticed yet, CAA and CAG both code for the same amino acid. So a codon, a single codon matches one amino acid, but one amino acid can have numerous codons. So if I look over here at leucine, leucine could be coded for with CUU, CUC, CUA, or CUG. Proline can be coded for with CCU, CCC, CCA, or CCG. So codons, one codon, one amino acid, many codons. So this also has to do with some evolutionary discussion. I'm not really sure. Evolutionary reasons. How about that? Yeah. So think about it. Remember we talked about mutations and how, like for example, Tay-Sachs is caused by a single base change. And so sometimes a single base change can be catastrophic. Like in Hayden's case, it was catastrophic. It happened to be at a very important location, and it completely changed the entire protein. So much so that the protein was never really even created. It was the wrong protein. But so there, over time, throughout evolution, we have developed ways to fight against mutations, to fight against kind of like safeguards. Yeah, we put safeguards in place to kind of give ourselves a backup plan. And so you could see how one single mutation in just one letter is not always terrible, because if CUU was the codon or just one of the sequences in a very important gene, if that third letter was the one to get mutated, let's say it mutated into CUC, well, nothing's really happened, right? Because it's still coding for leucine. It's still coding for the same amino acid. Or if it was changed to CUA, not really that big of a deal. It's the same amino acid. And so some mutations are more harmful than others, and others are really that don't even matter. There's really no change at all. The big changes would be if it happens to be at the start of the codon. If it changed from a C to an A, well, that completely changed your list of options. Completely changes your list. And so those would be a lot more impactful. Those would have a lot larger ramifications. Okay, so now we know how to transcribe and translate. Let's try it. So if I give you this template DNA sequence, TAC, GCT, AGT, ACG, ATT, I want you to transcribe this into a strand of mRNA. So remember, you're acting like RNA polymerase, adding those complementary base pairs. So go ahead and do that now. All right, drum roll, please. If you said AUG, CGA, UCA, UGC, and UAA, then you were correct. Give yourself a little pat on the back. So remember, T goes with A binds with U, because it's RNA, so it's uracil, and there should be no T's in any mRNA strands. G goes with C goes with G, so on and so forth. Now, I want to ask you to act like a tRNA molecule. I want you to read this strand in terms of their specific codons, because we're going to be matching up it with its anticodon. So all you're doing is separating this sequence into its corresponding triplets. So you're not going to add any complementary stuff. You're not going to make it opposite. The strand should not be opposite. I just want you to separate them into their triplets. Okay, do that now. All right, so if you said this, AUG space, CGA space, UCA space, UGC space, UAA, then you're a-okay. And so I'm just saying you're acting like a tRNA because you are just you're gonna match up your anticodon with its complementary codon. Okay. And so then I want you to translate. Now that we have our sentences, we have these codons, we need to actually read them and then act like a ribosome and then bind them, read them and bind together the specific amino acids. So I want you to go back onto, I want you to have your notes open and look back at your genetic code, and I want you to translate. I want you to look up what AUG, which amino acid is coded for by AUG. Look at CGA, which amino acid is coded for by this. UCA, which amino acid does that correspond with? UGC, which amino acid? UAA, which amino acid? And you don't have to spell out the whole word. You can just use the first 3 letters. So if I go back here and I look at leucine, you can just put LEU, or proline, you can put PRO, glycine, you can put GLY, alanine, you can put ALA, and so on and so forth. So you don't need to write out the whole word, but just the first 3 letters. Okay, so go ahead and do that now. All right, let's see if you were correct. So AUG codes for methionine, MET. CGA codes for ARG. UCA codes for SER. UGC codes for cysteine, and UAA is a stop codon. So then the ribosome would stop translating the message and would release the completed protein. Okay, and that is translation.